Okay, so we left off on Wednesday basically talking about some of the outcomes of the Human Genome Project, and specifically on Wednesday we left off talking about how or what evidence there is that our species interbred with a couple of other species within our genus, and then timing of everything makes things a bit interesting. So just to kind of put a bow on that discussion that we had on Wednesday, I don't think you can really argue that the humankind is only Homo sapiens, okay? Unless you're going to put some of these other species that it's very clear that they interacted with our species, both genetically and socially, as Homo sapiens as well, and say it's really irrelevant to classify them as a separate species. Unless you do that, there's really no way to say that the humankind is just our species, okay? And so what that leaves us with is the responsibility to kind of put this into the context of what Scripture teaches about human origins, okay? And so humans began as a single breeding pair, and so all of these other species of humans have to be post-Adam and Eve. And in fact, all of these other species of humans have to be post-flood. Okay? Because as far as timing of everything, the best, I think, the best model for flood, the end of the flood, is at the very end of the Cretaceous and the very beginning of the Cenozoic era, which in conventional dating would be about 65 million years ago. All of these species in our genus are from what would be the last 2 to 3 million years in conventional time, okay? And so we're talking about way even after the flood that we start finding these different types of humans, okay? On Wednesday we talked about that the amount of diversity, both genetically and morphologically, between our species and Homo neanderthalensis is fairly similar to the amount of variation present in just our species, both genetically and morphologically. But these are all going to be post-flood, and probably a long way post-flood, several hundred years, maybe even 1,000 years post-flood. Yeah, Jesse.
So is it possible that like all these different genomes that existed in those different, I don't know what you'd call them, but like types of human breeds would be like, they all got killed off in the flood?
It's possible, but if that's going to be the case, then they are being buried way after what we tend to consider as the end of the flood deposits. So, which means we need some mechanism by which we are preserving those bodies for what could be hundreds, if not even 1,000 years. And so that's tough. Bodies don't tend to stick around that long. I think it's a lot more likely that what we're looking at, and this is what Dr. Francis and Dr. Wood wrote a couple chapters about in the book, What Happened in the Garden. The best models is that these are all post-Babel. That these are post-Babel human groups that became extremely reproductively isolated. Not enough to fully isolate them from the rest of humans, because there's still some interbreeding, but enough to where their genome becomes unique, and they become morphologically unique. Yeah. Okay. I think that's it as far as that discussion is concerned. And again, just keep in mind that the amount of variation between our species and Homo neanderthalensis is not all that much different than the present amount of variation in all living humans. Okay? All right. So, Encode. In the key terms, I mentioned to you that Encode was a project put together with the intent of figuring out what does the 98.8% of the human genome do. If it's not coding, which after the Human Genome Project we realize only about 1.2% of the human genome is coding, that is, gets translated into protein. And so this project was tasked with figuring out what it does. Is it junk? It can't be junk, because that's way too much of the human genome to be junk. And so what is it? What does it do? And so here are some of the findings of Encode. First of all, nearly 80% of the human genome has a very obvious biochemical function. That is, About 75% of it gets transcribed at some point during the life of the individual. And so only about 5% of it still has an obvious biochemical function, but doesn't get involved or isn't transcribed. And some of these would be the repeated regions that we know that are involved in either telling SYN proteins where to bind as part of the centromere, or they're at the ends of chromosomes functioning as telomeres. And so about 80% of it has a very obvious, not very complicated to figure out biochemical function. And then so the other 80% of it, there's a question of like, okay, well, what does this other 20% do then? Okay, so if 80% of it we know it's this obvious biochemical function, the other 20% works in other ways, maybe in between as setting spaces in between these, or has some kind of regulatory role that we haven't discovered yet. All right, some other outcomes of the Encode project. Something we found during this project is that transcription factors, what they actually do oftentimes, it's not a transcription factor that's focused on a specific promoter. What these transcription factors often do is displace histones. They take the DNA that's wound around histones and they displace those histones to provide more easy access, thank you, Jesse, to that stretch of DNA. And when that happens, it basically becomes almost impossible to methylate, or even if you do methylate, to shut down that portion of the genome. So if you displace these histones, it is a way to make that an incredibly active site of the genome, to make it to where you can overcome what's happening at the level of the sequence. We already talked about this a couple weeks ago when I said that modifying histones can overcome the programming, the epigenetic factors on the actual stretch of DNA. Okay, and this is an extreme example of that. Now, we found that about 12% of the human genome is conserved among all humans, specifically for the purpose of maintaining proper three-dimensional shape of chromosomes and the proper location of those chromosomes within the nucleus. Now, this is not saying that only 12% of the human genome is conserved. Okay, this is not anywhere close. A much higher percentage of the human genome is conserved among all humans, but 12% of it is conserved for the sole purpose of maintaining proper three-dimensional shape, to make sure that the chromosomes condense properly, and so that the chromosomes behave properly. Now, due to alternative splicing, about 95% of the coding genes, those 21,000 genes, code for more than one protein, at least 2 proteins. Primarily due to alternative splicing, cutting out different sections of that immature mRNA as introns. Okay? And so it looks like there may be as many as 2 million different unique proteins in the human proteome. The proteome is the composite or the collection of all proteins produced in a human cell. There may be as many as 2 million of these produced from 21,000 genes. Okay? So you're talking about, on average, that gives us about 100 proteins per coding gene. Right? If my math is correct and it's Friday, so 21,000 times 10 would be 210,000 times 10 is now 2 million. Okay? 2.1 million. Okay? So you're talking about, on average, about 100 proteins from every single coding gene. Okay? And so it shows you that if that's the case, and it seems to be the case, we need more regulation than we ever thought was possible. Right? Because something has to determine how those immature mRNAs are going to get spliced. Okay? All right. Does that make sense? Questions? So the Encode Project, again, it was just like the Human Genome Project, was a group of entities given funding and tasked with figuring out some problem. Yeah, David.
Just a quick question. By trillion proteins, you mean for each individual coding gene? Is that what you're saying? Or?
About one, on average, there are 100 proteins produced from every single individual coding gene. Yes. Now, some of these are only coding for 2 proteins, right? There's 2 different ways to splice that immature RNA. And some of these are coding for thousands of different proteins, depending on how they're spliced. Okay? And then some of these, we've seen this, some of these are just going to be our different antibody types, right? That are actually alternative splicing in 3 different genes that then all get strung together as a single transcript. Okay. Last thing we need to talk about is how do we compare to other species? Okay, so we've spent some time saying, okay, according to the Human Genome Project, how much variation is there among living members of our species? And then we've spent some time talking about, okay, what does the other 98.8% of our genome do if it isn't coding? Okay, and a lot of that's going to be regulating the coding genes. But now we need to talk about, well, how does our genome compare to other species? So, comparative genomics allows us to do this, and it allows us to answer certain questions. 1, what are human-specific genes? And so comparative genomics allows us to do this because we can compare the human genome to the genome of other animals that are believed to be more closely related to humans or believed to be more further removed from the common ancestor with humans, and we can look at and see what genes which of these coding genes are found in the human genome and not found in any other species. Okay? And there are some unique human-specific genes. Also allows us to look at, okay, just what are the housekeeping genes? What genes are required regardless of what type of organism you are? What are the genes that are required whether you're a fungus or a plant, or an animal, or some single-celled eukaryotic life form, or even a bacterium? What are some genes that are required just to be a living organism? And a lot of those genes are going to function in processes that are present in all organisms. The production of ribosomes, glycolysis, the production of actin or something like actin is a cytoskeletal element involved in budding a single cell and pinching it off into 2 separate cells. This allows us to ask the question of what genes are necessary for multicellularity. What do multicellular organisms need that single-celled organisms do not need? And a lot of these are going to be regulatory genes. The ability to shut off coding genes in one cell but not in a different cell, so you can create that division of labor. Allows us to ask what questions are necessary for eukaryotes. In order to be a eukaryotic life form where your cell structure consists of membrane-bound organelles that isolate various reactions, okay, what genes are required to make that happen? Or what genes are required for mammals? Okay, we can ask all kinds of questions. Now, the nice thing about comparative genomics is it allows us to look and to see if we have this gene present in every single living form, it allows us to suggest this is a gene necessary for life to be possible. And then we have to ask ourselves the question of what maintains that gene in all of these various species. Okay? Regardless of what your view of origins is. Right? If your view of origins is that there are separate, discrete units of life created that have then diversified, or that all forms of life root back to a single source, regardless of what your view of origins is, you have to explain how are these genes maintained. Okay? If they're incredibly important, they're necessary, the proper protein product is necessary for life to work, what force is going to maintain those? Okay? And so we have different types of selection. We'll talk about them in this context. So the idea is, and again, regardless of your view of origins, the idea is that stabilizing selection is going to be the most important force for maintaining the particular code in those housekeeping genes, in those genes necessary for life. Because any mutation in one of those genes that distorts a protein necessary for life to work is going to be selected against by natural selection. Right? Make sense? If you get a mutation in a gene that codes for a protein necessary for life to work, natural selection should work very strongly against mutations in that gene. Because any mutation in that gene is probably going to result in an organism with extremely reduced fitness or an organism that just doesn't survive at all. Okay? And so stabilizing selection is going to maintain that. Meaning, even if you have discrete units of life created, where they were created as separate entities and then have diversified, there's still, the reason why we are still similar to them is because those genes are necessary for life, and natural selection works very hard to eliminate mutations in those genes. All right, now, if you're going to start to say, okay, well, how do we get differences among species, even within a created kind? Even if we have discrete, separate creations, okay, that then diversify after the creation event. Let's say some things that I think have very little controversy. Like if at the beginning of creation there was one type of dog that is then diversified into all of the species of dogs that we have. Okay, that's one that doesn't create a lot of controversy, because whether you're talking about a coyote or a wolf or a domestic dog, they're almost all still capable of interbreeding and forming viable offsprings. Now there are some, like foxes, that are a little bit more different, but then you get work on foxes where it's like people select for tameness in the foxes, and all of a sudden they start to look a lot like domesticated dogs. Their ears round out, they start to bark. They stop looking like foxes where it's like it's all gray or it's all red. They start like having these patches show up that is common in domesticated dogs. And so if at the beginning of creation there was a dog kind, and then it has diversified into our different species of dog, what force was the most likely culprit of separating those into individual species? And so this comes out of the textbook that positive selection is the best force for explaining the differences among species. That positive selection, that is when natural selection is selecting for an extreme, that's going to be the most powerful force. I added in here and/or discontinuity, talking about that. What the Bible seems to describe is that there are discrete kinds of life from the very beginning. And so some of that, a lot of those differences are in place as soon as these organisms show up. This, I think, is obviously a great way to describe why are organisms different. Well, because they were created separately. They were never united, other than in the fact that they were created by the same creator and from the same materials. This is a huge issue to explain even the differences among species within the same created kind. Because time is a huge issue with positive selection. We've talked about this a little bit already, and we'll see this a lot more as we progress through this semester. Natural selection is super duper slow. Super duper slow. Whether you have millions of years to accomplish a splitting of a species into two, or you have only dozens of years in a young Earth model, natural selection is not going to do it for you. Even millions of years is not enough time to set two species apart. It's just not. And we'll see why that is. It just, natural selection takes way too long to fix even if you gave a mutation a huge fitness advantage, something that would be almost beyond the realm of biological possibility. Like you've got this mutation and it gives the organism a 50% increase in fitness, which is just absurd. It would still take way too long to fix the amount of mutations that you need to differ organisms. So then the question is, okay, if positive selection is not going to do it, right? Discontinuity is going to be a great explanation among different created kinds, but what about within a single created kind? If it's not positive selection, what is it? And which force of microevolution works really quickly and especially when populations are small? Genetic drift. Random genetic changes. Okay? And so this is what the textbook says, although they don't talk about discontinuity because anyways, it would probably make sense why. But positive selection, I think, is going to be a very poor force to choose because it's just way too slow. Drift, on the other hand, drift is fast, especially in small populations. In a matter of just a handful of generations, drift can easily isolate a single species, a single population into two. Oh, yeah. Does it make sense? Okay. We find about 5% of our genome is conserved with all mammals. So about 5% of our genome, and this isn't just of the coding genes, this is 5% of the entire genome. Keeping in mind that we really only have 96.5% of the human genome sequenced, right? Because about 3.5% has yet to be sequenced. But about 5% of the human genome is conserved among all mammals. Okay, these are the mammal codes. This is what's required to be endothermic. Okay, this is what's required to produce the types of glands that you find in mammals. This is what's required to regulate the development in a mammal consistent way. Okay, these are the genes that are required to produce fur, if you have any. I have to share you a story. Really, it's not related to any way in this class. So I was allowing my hair to grow back just for fun, and a little change. And I was sitting on my daughter's bed reading to her, and she's playing with my hair. And she's like, "Daddy, I'm so happy your hair is growing back." And then she said, "But you've got all these bald spots. What are those all about?" And I was like, "Dang it!" So I shaved my head that night. So, all right. Sorry, that again is nowhere related, but I thought I would share that story with you so you can understand my struggles that I deal with as trying to lead my children. All right. Now, another 4% of the human genome is conserved as human-specific genes. So now we have 12% that we talked about before, conserved just to maintain the proper three-dimensional shape of chromosomes. 5% conserved because we have certain things as mammals that we need to do. 4% as human-specific signatures. We didn't talk about how much of it is conserved as eukaryote-specific sequences or how much is conserved as multicellular-specific. And it's because we haven't sequenced enough organisms' genomes to know that yet. We haven't. We've sequenced a lot of mammal genomes. We've sequenced a lot of human genomes. We've sequenced a ton of primate genomes, but just some of it we don't know yet. We don't know yet. All right. So we oftentimes hear that humans and chimpanzees differ. The human genome differs from the chimpanzee genome by a factor of 1%. That we are 99% similar to chimpanzees. If you just look at coding regions, that number is still greater than 1% variance. The 1% was, again, it's based off of only single nucleotide polymorphisms differing the coding regions. If you allow for copy number variance as well, this number goes as high as about 4% just in the coding regions. Okay? If you allow single nucleotide polymorphisms and copy number variance as you try to compare these two genomes, it's about 4% in the coding regions. And it's hard to know how much of it is in the regulatory regions. Because the chimpanzee genome, we do not have as many of them sequenced as we do the human genome. And the way it was done was just kind of, and the way it was reassembled was different, bless you, than the way we reassembled the human genome. It was basically reassembled by matching it to sections on the human genome. So you built all these, you did all these fragments like you do when you're sequencing something really big, the way you should do, and then it was realigned using the human genome. Okay? And so it's hard to know, but the number's probably more like 70% when you compare the entire genomes. Our genome is probably 70% or so conserved with the chimpanzee genome. So now you're talking about something very, very different than sharing 99% homology with chimpanzees. Okay. Because you're like, that doesn't make any sense if what you told us on Wednesday, Dr. Ingold, was that variation among all living humans may be as high as 2 or 3%. And we're only 1% with chimpanzees. It doesn't make any sense. We're not even 1% in the coding regions. We're 1% if all you account for are single nucleotide polymorphisms. And then if you also allow for copy number variants, we're more like 4% in the coding regions. And probably more like 30% genome-wide. All right? Any questions about that? So again, a lot of this we're just kind of waiting. We need more people to sequence more genomes from more animals, to sequence more genomes from chimpanzees. Yeah.
Why, when they didn't, okay, so if you don't need like some other reference genome to sequence like human genes, why did they think?
Because it costs less.
Costs less?
Yeah, I mean, you've done this before, right? You've done sequencing where you're just trying to piece together something that's like maybe 1 million bases long, or you watch people do this, right?
Yeah.
And work for days and days and days to patch something back together that's only maybe 1 million bases long. It's expensive because you're paying for computer time, you're paying for people. It's an expensive process. But to just attach it to an already sequenced genome, and instead of trying to figure out where all these fragments fit together, it takes way less time and costs way less money.
That doesn't make sense though, you're not sequencing their genome, you're just putting it in cells.
Well, the idea is you have sequenced everything. You've sequenced everything. Remember, so shotgun sequencing and next generation sequencing has a lot of back-end analyses, right? Because you're taking and you're basically, you've got fragment 1 and fragment 2 and fragment 3 and fragment 4, and you're looking for these places of overlap, okay? But the problem is this section here might overlap with this, but it might also overlap with this. It might also overlap with this. It might also overlap with something like this. And so now you're having to do an enormous amount of analyses to figure out where does it overlap with for sure, and where does it not belong with, right? Because there's multiple ways that this puzzle can be put back together. But if instead you're like, we're not going to do this because that's going to be very labor intensive and very expensive, what we're going to do is just take all of these chimpanzee fragments, okay, all of these fragments that we've sequenced, and we're just going to figure out where do they fit on the human genome with a certain level of homology, right? It's similar to what we do when we do blast searches, right? And it comes back and you're like, okay, it matches 75% of this published sequence. You're like, cool. All right, it probably is that portion of the chimpanzee genome. It's just less work than trying to put all the pieces together. You basically are like, it'd be akin to, you've got a jigsaw puzzle, right? And instead of figuring out where all the pieces go together, you're just like, okay, this piece, you take the box and you're like, this piece looks like it's this part, and you just set it on the box there, right? You're not trying to put all the pieces together, you're just like, what part of the picture is it probably like? And you're like there, and you're like, how similar is it to that picture? That part of the picture. Yeah, it's pretty similar. Well, what's pretty similar? You're like, well, they only differ by 5 bases in, 100 bases. You're like, okay, so now, and then you just do all these pieces. It makes it a little bit simpler, a little bit less labor intensive. David.
Maybe more of a biological question and more kind of a theological question.
Yep.
So, is it the responsibility of the individual to try to understand these things, or the responsibility of kind of leaders in the scientific community, especially in the Christian sphere, to kind of assist Christians in understanding this material or to make it very plain and easy? Because things like sequencing of DNA, it's something that most people would just like, go right over their head.
Yeah, I mean, it's like anything else. the methods are always going to be harder to understand than the implications, right? The implications are usually something that is fairly easy to understand, right? So if I tell you, I don't tell you how I did the analysis, right? But all I tell you is that in just our like coding genes, right? Just the parts of our genome that we know make something, right? Make an obvious protein product, that we are 96% similar to chimpanzees, right? I don't need to tell you anything about how I did that for you to say, okay, the implication of that is that we share a lot of similarities with chimpanzees, right? And then you have to start to think about, well, what does that actually mean, right? What does it actually mean? Is that something that we wouldn't predict? And what's interesting is, up until maybe until Linnaeus did a really good job of creating a taxonomic system to explain the diversity of life, and at that same time that all these exploratory groups were doing a really good job of getting specimens from all over the world, and we were starting to get a really good sense of what organisms had lived in Earth's history based on fossils. Up until that time, most people assumed that anything that you could imagine to exist would exist. That God is capable of creating basically anything. And so if you looked at every organism alive, you would see a complete, perfect, filled-in spectrum, even with separate, discrete creations. It wasn't until, probably the 19th century to where we started to say, no, like we need to find very obvious places of discontinuity where these things don't exist, and that is our best evidence for a special creation, right? So, all that to say, I don't think anybody should be surprised that there should be some animals that are more similar to us than others. Because the reality is, there's no reason why that shouldn't be the case. There should be animals that are more similar to us than others, right? We're apex predators. We have very complex social societies, and we invest a lot of effort in developing the next generation. We invest a lot of parental care. You should expect that in animals that invest a lot in parental care, that are apex predators, that have complex social societies, they should be more similar to humans than animals that don't have those. I don't know if that answers your question.
Okay.
So I would say, anyone who has an understanding of how you do these analyses has a responsibility to explain it to others. If you have any kind of an understanding of the methods behind how we do these analyses, then you have a responsibility to help people to understand the implications of saying things like, we are 99% similar to chimpanzees. Well, now you have a responsibility to tell them, well, that's actually not true. That's only true if you only use one type of difference. If you use all of the types of difference, it's more like 4%, we're 96%, but that's only in the parts that actually code for protein. And you're like, really, it's probably more like 70% genome wide. Yeah. So.
I think it's just kind of trying to, especially nowadays where the implications are made on assumptions generally.
Sure.
That it's declaring that there's a bias here is really important.
It's going to be really interesting to see what things are like in 20 years. Because in the last 20 years, the scientific community, so scientists that are my age and a little bit older have become very dissatisfied in the traditional explanations of macroevolution. Because because natural selection is way too slow. It's way too slow to fix these differences, because we don't have a great mechanism by which this can happen rapidly enough in the amount of time these organisms have been around, and because the fossil record is just rich with discontinuity, with groups that all appear at the same time with no way to connect them. Scientists that are my age and even a little bit older are very dissatisfied with the neo-Darwinian explanations of universal common descent, and they're starting to abandon that as an explanation with no other explanation. So, which will be interesting to see what it's like in 20 years, because people that are my age and a little bit older are the ones that are now getting tenure at big universities and are shaping the next generation of scientists. And so, yeah, it's just going to be interesting to see what it's going to be like in 20 years. So, I don't know. All that to say, what's that? Yeah, I mean that's a popular explanation now that several different types of life showed up simultaneously at the same time on Earth because they were brought here from somewhere else. That was Crick's original idea, and it's gained some popularity. All right, let's do this.